So we're in the realm of plant biology still, but now we want to have a look at transport in the phloem of plants. So our last discussion was around the xylem, and now we want to have a look at the phloem. So let's look at what the phloem is to start off with. So phloem are vessels which transport organic compounds around the plant. And these organic compounds that we talk about are carbohydrates, which we know are just sugars. And if you've ever had a look at a tree and seen this sort of sticky substance oozing out of it that we call sap, that is what is usually contained inside the phloem vessels. Now, when we talk about the transportation of carbohydrates around the plant, we talk about this direction of movement as going from the source to the sink. So let's have a look at what that means. So the source refers to the site of production of the carbohydrates, whereas the sink refers to where the carbohydrates are needed. So looking at those definitions in more detail, we know that photosynthesis occurs in the leaves of plants. So carbohydrates are being produced at the leaves, and therefore leaves are typically the source that we're talking about. So they'll be transported from the leaves, which are the source, and then they're going to what we call the sink. So let's have a look at what the sink refers to. So we know that the sink is where carbohydrates are needed, and so if we look at the site of production of carbohydrates being in the plants, that's all well and good, but we know that the stem and roots also need some carbohydrates. They also need to be fed. And so therefore the stem and roots are typically the sinks that we are talking about. So when we're talking about from source to sink, we're usually talking about from leaf to stem or from leaf to root. So that's like the sort of basic definition of a source and sink. Let's consider tuber plants though. So tuber plants refer to plants with modified roots that can store carbohydrates. So the ones that we know and love are probably potatoes, maybe beetroots and carrots. A bit less familiar are yams, but these are all types of plants where the roots have been modified to be able to store carbohydrates. So that potato that we eat is actually a modified root that has stored excess carbohydrates from the potato plant. So carbohydrates we know are produced in the leaves, as we've just discussed, and then they are sent to tubers to be stored. So our leaves would be the source, and then in this case our tubers would be the sink, because that's where the excess carbohydrates are being sent to. Let's have a look at other conditions though. If the plant is under stress conditions and is under conditions where it's unable to photosynthesize at a sufficient rate, for example, during autumn where there's a reduced number of leaves, or during winter where the period of sunlight is a lot less than in summer, in such conditions, carbohydrates are then taken out of storage. So they're taken out of, for example, the potato and then sent to other parts of the plant. So in this sort of situation, our tubers are the source. So typically in ideal situations, they are the sink, but in conditions such as being the season of autumn or winter, or in other stress conditions, we need carbohydrates to be taken out of storage because our leaves are not producing enough. And so therefore in that situation, our tubers would be our source. So just to summarize what we've looked at, we've got a table here comparing the source and different sinks. So leaves are our main source, and then we've just discussed that tubers can also be a source when we're in non-ideal situations. Sink, we know that tubers can also be a sink, so that's why that's in both columns, but we also have our typical sinks being our stem, our roots, and two others that we haven't really discussed, but you should bear in mind are fruits and seeds. So these are sinks that receive carbohydrates. So remember, this all takes place, this transportation of carbohydrates around the plant takes place via the phloem. So let's have a look a little bit at the structure of our phloem vessels. So phloem are composed of living cells, and if we compare this to our xylem, which we've discussed, we know that xylem are composed of dead cells. So hopefully as we're looking at the phloem now, you'll be able to revise over your xylem understanding and see some similarities and differences. So phloem are composed of living cells as opposed to dead cells. And in phloem vessels, there are two main cell types. We have the sieve element cells and companion cells. So our sieve element cells are our main column of the phloem, where the transportation of carbohydrates takes place. Our companion cells sit on the outside of our sieve cells, and they support our sieve cells and provide particularly metabolic support to the sieve element cells. So sieve cells are connected via sieve plates, and we'll have a picture of this in one of the following slides. For now, just understand that the individual sieve element cells are connected via sieve plates, which have these little holes or pores in them. So they are porous to allow for that flow to continue between cells. Sieve element cells have a cytoplasm, but they have no nucleus, and they also have a reduced number of organelles. And this is in order to maximize the amount of space available for the transport of that sap or the transport of carbohydrates through the vessel. Let's look at our companion cells. So we know that they provide metabolic support for our sieve element cells, and this is because the sieve element cells have no nucleus and they have no organelles, or they have reduced number of organelles, and therefore they need metabolic support. So that's what our companion cells provide. Companion cells also facilitate the loading and unloading of materials at the source and at the sink, and we'll have a look at this in a bit more detail. We also know that companion cells have many mitochondria because they need to fuel the active transport of loading and unloading, which once again we will look at in the coming slides. Sieve element cells are unable to sustain independent metabolic activity, and so that's why we need companion cells. So that's sort of the main thing to take away. So we have our sieve element cells, which are the main column in which transportation takes place, but we know that they are unable to sustain independent metabolic activities, and so that's why we also have these companion cells. So let's look at this picture that I drew quite poorly, but it sort of just shows us these key elements of our phloem. So we have our sieve element cell, which is the main column in the middle. And we can see that represented in purple, it has a reduced number of organelles, and it has no nucleus. So we can see in our companion cells, which are the skinnier cells on the outside here, these the green dots in the center, they represent a nucleus. The purple represent organelles, and so we can see our companion cells have a nucleus and they have more organelles than that of our sieve element cells. Now between each sieve element cell, we have these little plates that are perforated, so these white dots represent perforation, and this allows sap to flow through the main column of the sieve element cells. So that's the overall structure. So let's briefly compare xylem and phloem in terms of their structure. So we know that xylem is a continuous tube, so it has no end plates, whereas if we come over to our phloem vessel, we can see those plates with perforations that separate the individual cells. We also need to remember that xylem are composed of dead cells and phloem are composed of living cells. And if we look at our xylem here, because they're composed of dead cells, they also have this thickening around them called lignin, which stiffens the wall up. And on the other hand, if we look at phloem, it doesn't have lignin around it. The other key thing, obviously, to understand is that xylem transports water and the minerals that are dissolved in water. So when we talk about minerals, we're talking about simple ions such as potassium, sodium, and so just water and just the sort of salt that is dissolved in water. Whereas in our phloem vessel, we've got yes, we've got a little bit of water, but it's mainly food, and so that water is there to just turn our carbohydrates into a sap, into a liquid, so that it can transport throughout the plant. The other thing that we haven't really discussed is that in a xylem vessel, the direction of transportation is one way, so it's unidirectional, whereas in a phloem vessel, it's bidirectional. So we need to be able to identify xylem and phloem in microscopic images. So we need to be able to see, okay, which is the xylem, which is the phloem. So the first thing to note is when we look at roots, we need to remember that the roots are the part of the plant that go into the ground, and they have a much smaller diameter than the stem, which is in between the roots and the leaves. So the stem has a larger diameter, whereas the root is quite small and hair-like. So roots, being small in diameter, they only have one central vascular bundle. And when we talk about a vascular bundle, we're talking about the bundle that contains xylem and phloem. So we have a bunch of xylem and phloem enclosed in a bundle, and we call that bundle a vascular bundle. And in a root, there's only one bundle, whereas in a stem, because the stem is larger in diameter, it has several vascular bundles. when we want to distinguish between xylem vessels and phloem vessels, we need to understand that the xylem are usually located more internally, whereas the phloem vessels are located more externally, and this is true of both roots and stems. Some special notes for identifying and distinguishing xylem and phloem in the root is that in the root, the xylem vessels appear as a cross. So one way to help you remember is to think X for xylem. So where you see that sort of X shape, we know that that's made up of xylem, and the phloem are situated in the surrounding gaps. This is for root, images of a root. And then for a stem, the thing to note is that the xylem, once again, is located internally and the phloem externally, and they're separated by this layer of cells called cambium. So we're going to have a look at some microscope images of both stems and roots in order to have a bit more understanding of these points that have been raised. So remember, the root has one central vascular bundle, whereas the stem has several vascular bundles. So based on that information, are you able to identify between these two images which one is the stem and which one is the root? So let's have a look at this first image here. So we can see that this is a root cell because there's just this one central vascular bundle. Now, if we think about the xylem rule and thinking about that X, we can see that we've got this vague X shape here, and so we know that these highlighted cells would be our xylem vessels. And just to orientate you to this image, this is a picture of a root, and then just imagine cutting a blade through that root, and then we're looking at the cross section. And so that's what we see here. And so highlighted, we have our xylem vessels, and then remember in the surrounding gaps, we've got our phloem. So back to the blank image, can you now see where the xylem vessels are and where the phloem vessels are on the outside? Now this is our stem. And so in the middle here, where it's white, this is what we call the pith. So there's really not much going on here that we need to know about. But if we look to the periphery, we see these, there's several vascular bundles. And I'm not too sure how many there are, probably about 20 or so. And so we've got these vascular bundles. And remember, the main rule is that xylem are located more internally and our phloem are located more externally. So are you able to identify which ones are our xylem? So hopefully you were able to identify that these were our xylem, so highlighted here in yellow. We've got the xylem from each vascular bundle. Then we've got this layer in light grey of cambium, and then on the outside, these darker, almost black circles would represent our phloem. So if we go back to the unhighlighted image, are you able to see where the xylem is and where the phloem is? So once again on this slide, there's just some notes and some steps that you can go through when you need to identify xylem and phloem. So you need to ask yourself, how many vascular bundles are there? If there are many, then you know you're looking at a stem, but if there's only one, you are most likely working with a root. The main thing is that xylem are situated internally, whereas phloem are situated externally. And then one thing that we haven't discussed is that xylem have a larger diameter than phloem. And so the circles for xylem should be bigger than that of phloem. And we can see that in this image because remember how I said the phloem sort of appear to be black? That's because of how small they are in diameter compared to the xylem. And so they appear to be more dark in color. If we go back to our root, the same is true. So we've got our xylem with larger circles, and then our phloem on the outside with smaller circles because our phloem are smaller in diameter than our xylem. Let's have a look at this principle of phloem loading. So phloem loading is an active process in which sugars are loaded from the source into the phloem and then transported down through the phloem, and then they are unloaded from the phloem into the sink. And so phloem loading requires energy as we are going against the concentration gradient, and we'll unpack this in some more detail. So here we have another poorly drawn image of a phloem, and then these light green squares representing cells of the leaf and cells of the root, or a cell of the leaf and a cell of the root. So this top one here is a cell from the leaf, so it would be our source cell. And then this bottom light green square here is a cell from the root, and so it would be our sink cell. Remember, we have movement occurring from source to sink. And so from the leaf or from the source, we need these orange dots here, which represent carbohydrates. We need them to go into the phloem vessel, which is indicated in dark green. And so this is all well and good, but we can see that we've got more carbohydrates already in the phloem than that is in the cell, in the leaf. And so we know that in nature, we tend to move in the opposite direction. So we like to go from high concentration to low concentration. But what we need here is the movement from low concentration in the leaf to high concentration in the phloem. And so because this is going against the concentration gradient, this is an active process and will require energy. Once this active transport loading takes place, the carbohydrates will go down through the phloem and then passively transport into the root. So the key thing to note is that active transport is required to unload carbohydrates from the source into the phloem. This is another image from BioNinja that shows this, and then it adds another layer of detail. So that layer of detail is that as carbohydrates are actively transported from the source into the phloem, due to that increased concentration of solute in the phloem at the top, we're going to have a transpiration of water into the phloem, because remember, water chases solute. So the key thing to note is that xylem and phloem are situated side by side or in close proximity. So where we have an increase of concentration of solute in the phloem, we're going to have water traveling into the phloem. Then as it goes down, as the sap goes down through the phloem via translocation, it's going to enter into the sink, and so it's going to decrease in concentration, and therefore water is going to leave the phloem and enter into the xylem. The other thing to note is our companion cells. So we know that at the top we have active transport to unload from the source into the phloem. And one thing to note is that our companion cells, because they are our metabolic companions, they provide the energy to facilitate this process. Now we can also measure the rate of phloem transport, and we use an insect called aphids. And aphids are an insect which feed on sap from the phloem. So they project their stylet into the plant's sieve tube to extract the sap. So that protruding mouthpiece that they have is called a stylet. And if we have a look at this image here, if we look at this actual image, we can see this protruding structure entering into the leaf. And then if we look at the diagram, we can see that this stylet pierces the epidermis, it pierces the mesophyll, and goes right down, even piercing the companion cell, going right into the phloem to drink the sap straight from the phloem. This image is also from BioNinja. So if the aphid is severed from its stylet, the sap will continue to flow from the plant into the stylet, into that mouthpiece. So aphids can be used to collect sap at various sites along the plant's length, and then we can use this data to measure the rate of phloem transportation. So how this is done is that the plant is grown in conditions where it is exposed to radioactive carbon. So it is exposed to marked carbon or labeled carbon. So the leaves, by photosynthesis, will convert this radioactive carbon into radioactive sugars. So the reason why we radioactively label the carbon so that it is transformed into radioactively labeled sugars is so that we can keep track of where the sugar is, because we can see the radioactive labels using technology. So after exposing the plant to radioactive carbon, and then the plant converts this carbon into sugars, we position aphids along the plant's length and encourage them to feed on the phloem sap. Once the feeding has commenced, the aphid stylet is severed, and then sap continues to flow from the plant at the selected positions. So the sap via the stylets is then analyzed for the presence of radioactively labeled sugars. And then we can use this data to calculate the rate of translocation, or the translocation rate. And so it can be calculated based on the time taken for the radioactive isotope. So it can be calculated based on the time taken for the radioactive isotope to be detected at different positions along the plant's length. So if we've got a lot at the start, a lot of sugar at the start, but not much at the end, we know that the rate of translocation is quite slow. Whereas if we've got not much at the start, but we've got a lot of sugar at the end of the plant, we know that the rate of translocation is fast because it was able to travel to the end faster. That was a very layman's way of explaining it, but obviously we use numerical data and calculations in order to figure out an actual rate. So that brings us to the end of our presentation about phloem and transportation in the phloem. If you have any questions, please let me know. Just email me and I'll get back to you as soon as possible.